Hi guys, in this class let us communicate in the language of insects. There are various ways by which as a human we communicate with each other, like speaking language, sign language or body language, etc. Insects communication is more advanced than ours. They use chemical languages, chemical substances that mediate communication between organisms are called as semiochemicals. Semiochemicals can be classified into two types, pheromones and allelochemicals. Pheromone is a semiochemical that is used for communication within the individuals of same species. Pheromone is secreted outside in the environment that causes specific reaction in the receiving individuals of the same species. Based on the responses elicited, pheromones can be classified into two groups, primer pheromone and releaser pheromone. Primer pheromones trigger a chain of slow and steady physiological changes in the receiving individuals without any immediate change in the behavior. They act through taste sensilla. That means individuals of the same species has to taste this pheromone in order to react. These primer pheromones are mostly in greater use for the social insects. Caste determination and reproduction in social insects like ants, bees, wasps and termites are mediated by primer pheromones. These pheromones are not much practical value in integrated pest management. Our second kind is releaser pheromone. These pheromones produce an immediate change in the behavior of receiver. Unlike primer pheromones, releaser pheromones do not need to be tasted in order to react. Most of the releaser pheromones are absorbed by the sensory organs in the antenna, so they are quick in responding to the releaser pheromones. Releaser pheromones are subdivided into four major categories based on their biological activity. Let us see them one by one. First one is sex pheromones. They are released by one sex only and trigger behavior patterns in the other sex that facilitate in the mating. They are most commonly released by females but may be released by males also. So far, 150 species of insects, females have been found to release the sex pheromones, whereas only 50 species were recorded for males to produce the pheromones. Following are some of the female sex pheromones identified in insects. Like in case of silkworm Bombyx mori, it is bombicol. In case of pink bollworm, it is Gossypol. In tobacco cutworm, it is spodolure or litlure. In Helicoverpa armigera, it is helilure. In case of honeybee queen, it is queen substance. Some examples of male insects producing the sex pheromones are cotton bollweevil Anthnomus grantis, cabbage looper Trichoplesianae, and Mediterranean fruit fly Ceratophrys capitata. These are all the few examples where the male insect produces the sex pheromone. Our next kind is alarm pheromones. Alarm pheromones are chemical substances that is released to warn the members of the same species. The warning is mostly about the presence or attack of the predator. It is produced by both male and females of the population. Alarm pheromones have been reported in Hemiptera, Isoptera and Hymenoptera. There are various organs involved in the production of alarm pheromones. Like in case of aphids, it is cornicles. In termites, it is produced by the cephalic glands. In case of worker bees, it is sting glands. In ants, it is Duffer gland, anal and the mandibular glands. Our third kind of pheromone is trail marking pheromone. Every one of us would have observed the ants marching in the line. When we do any interruption in between the line, ants will be roaming like they are lost in somewhere else. Why? The reason is trail marking pheromones. These pheromones are laid in the form of continuous lines on the soil. This trail is perceived by the antennae of them to reach the destination, which could be a mate or a food source. Trial pheromones help in finding mates and foods efficiently in social insects. It helps to maintain the social integration and cohesion inside the colony. The organs that is involved in the production of trial pheromones are sternal glands in termites. When we see it in ants, it is Duffer glands, pollen glands, poison and rectal glands. Trial pheromones can be used to attract and kill ants, which is used in case of the baits of ants. So we can use these trial pheromones to attract and kill the ants. Our next kind of pheromone is aggregation pheromone. Have you seen some of the insect species aggregated in certain place for certain period of time? This is due to aggregation pheromones. These pheromones induce the aggregation of insects in certain place. The reason for the aggregation may be for the protection against the natural enemies or reproduction and feeding or combination of all these reasons. They are released by either male or female, which attract both male and female of the same species. Aggregation pheromones are mostly known in Coleoptera and Dictyoptera. Example for these aggregation pheromones are ferrolure of red palm weevil. So, pheromones are intraspecific semiochemicals. That means they are involved in the chemical communication inside the same species. Our next semiochemicals are Allelochemicals. These Allelochemicals are involved in the communication between two different species, such as insect and plant interaction. So, these semiochemicals are going to be produced by one species, which will trigger response in the another species. The response could be positive or negative for the emitting species. If the chemical produced is positive for the emitting species, it is called as Allomones. Some of the examples for Allomones are Gossypol produced by cotton is toxic to the cotton bollworms. And some of the wild potato species repels aphids by releasing the aphid alarm pheromone, that is pheromones. Pentadecanale in rice inhibit the growth of brown plant hopper. So, the allomones are intraspecific semiochemicals that favors the producing species. So, the repelling chemicals are allomones. Even the sting glands present in bees are also coming under the category of allomones. If the chemical emitted by one species produces negative effect on it, it is called as Kairomone. These Kairomones are favorable to the receiving species. The chemicals released by the insects feeding on plants serves as a clues to their natural enemies. For example, the aphid odours are oviposition stimulant and arrestant for the predator syrphid fly and attractant for cecidomyiid predators. So, these aphid odours stimulate the syrphid flies to lay their eggs nearby the aphids and arrest the movement of predators near the aphids to feed on them. The larval parasitoid of diamond bag moth, that is Epondylus plutellae, finds its host by the combination of diamond bag moth and its host cabbage. When the chemical emitted is favorable for both the emitter and the receiver species, it is called as cyanomones. Some of the plants emit cyanomones, which is going to be attractive for both the pest and their natural enemies. For example, the corn seedlings attacked by the Spodoptera exigua releases a large number of terpenoids, which serves as clues for their parasitoid Codicea species. The odor emitted by the corn seedlings attracted both their pest and their natural enemies. It is called as cyanomones. When the chemical substances emitted by a non-living material trigger the reaction which is going to be favorable to the receiving organism but detrimental to an organism of another species which may be found in or on the non-living material. These kind of chemicals produced by the non-living material is called as apnemones. Examples for apnemones are the odor of fish meal attracts the flies. So the sorghum shoot fly can be controlled using the fish meal trap. The smells of fermented molasses attracts the Spodoptera litura, which is used in the poison baiting in order to control these pests. And also there is another chemical substances which is going to be detrimental for both the receiver and the emitter, which is called as antimones. Let's see how these pheromones are used in insect pest management. After identification of the natural pheromones and their chemical nature, the synthetic analogues of them are manufactured and used in the insect pest management. These synthetic pheromone lures are available for following pests. Like in case of Helicoverpa armigera, it is available in the name of Helilure. In tobacco caterpillar, it is called Spodolure or 4-in-SL. In case of pink bollworm, it is Pectinolure or Gossypol. Likewise, it is also available for rhinoceros beetle, red palm weevil and spotted bollworm. So, with these pheromones, pheromone traps are made. There are two types. First one is the sticky trap or the delta trap, which is mainly used for the pink bollworm trapping. Second one is the bucket trap, which is used for red palm weevil and rhinoceros beetle trapping. In the delta trap, the pheromone is placed in between the glue surface so that when the insects are attracted to the pheromone, they are going to be trapped in that. In the bucket trap, the pheromone is going to be placed inside the bucket, so when the red palm weevils are attracted inside, they are going to be falling inside where the insecticides are placed. Among the different kinds of pheromone, the sex pheromones are highly used in pest management. These sex pheromones can be used in three ways. First one is monitoring or survey trapping, where the sex pheromone can be used to trap the opposite sex of an insect, which can be used to monitor the pest population in the field. This is used to detect the pest infestation in early stage and to take management practices at appropriate time. The next kind is by mass trapping. In monitoring, we are going to trap the part of the population to identify the entry of insect. But in case of mass trapping, we are going to trap the entire population, mainly the males, and reduce the number of males for mating. Pink bollworm can be successfully trapped using the pheromone Gossypol in sticky trap, that is delta trap. Spodoptera litura and Helicoverpa armigera can be trapped using the funnel traps. The third way in which sex pheromones are used is as a mating disruption or male confusion technique. Here we can see in the normal condition, male can easily locate the females using the chemical clue from the female, that is a pheromone. But when we spray the same chemical all over the plants, it cannot locate the female, leading to the failure in mating. This technique involves widespread application of synthetic pheromones over a target crop, just like we spray the insecticides. The insects are unable to locate their mate using their own pheromone. As the synthetic pheromones are sprayed all over the crop, the insect cannot locate their mate. Ultimately, this will reduce the mating possibility and leading to the elimination of the population. This was successfully demonstrated in case of pink bollworm in cotton and yellow stem borer in rice. When we see the advantage of using these pheromones, they are non-toxic and eco-friendly as comparing them with the toxic chemical pesticides. They are species specific and safer to the natural enemies. When we look into the disadvantages, the effective pheromone availability is limited to a few species and it is not available for all the pests of the crop. Many pheromones are expensive to manufacture. Some are unstable and lose their effectiveness within a short period of time. And isolation, identification and synthesis for their commercial exploitation requires specialized skills, as it is not available in plenty. So, the exploitation of these pheromones for every pest is not yet possible. Chemicals that make insects orient towards their source are called as attractants. These are all the chemicals present in the host that attract or lure insects for feeding. They stimulate the olfactory receptors of insects usually present in the antenna. Some of the natural and synthetic products are useful in pest management as effective food lures. They can be used to attract the pest and kill them by employing some other means of pest control like poison bait. Some of the examples of attractants used in pest management are methyl eugenol and trimethyl, which is used to attract the males of fruit flies, luring them from over 1 kilometer downwind. Moths and butterflies are attracted towards fermenting syrups, sugars and molasses. The metaldehyde is used as attractant for snails and slugs, which is highly utilized as the baits in greenhouse conditions. So these attractants are used to make the bait where the insecticides are applied so that insects are controlled using the poison baiting. Likewise, a chemical that attracts the adult female and helps to identify the oviposition site is called as oviposition lure. A surface sprayed with these chemicals will attract the insect to lay their eggs on it, which can be destroyed. Some of the examples for oviposition lure are para-methyl-acetophenone, which attracts rice stem borer to oviposit, and the extracts of corn attracts the corn earworm Helicoverpa armigera to oviposit on them. So, using this lure, the insect can be attracted towards them to lay their eggs, later which it can be destroyed. These semiochemicals can be easily integrated into the integrated pest management so that diversified pest management strategies will lead us to control the pest effectively rather than going for the chemical pesticide treatment alone. Okay, let us move on to the question part. Difference between Allelochemicals and pheromones. Right or wrong? Pheromones are used in intraspecific communication and Allelochemicals are used in interspecific communication. Question number 3 is, in the view of receiving organism, mention whether the following chemical is positive or negative or both. Allomones, cyanomones, Kairomones and epnomones. Number of traps required per hectare in case of monitoring is. Write down the sex pheromones of following insects: Spodoptera, Helicoverpa and pink bollworm. Use of mating disruption in pest management. Question number 7 is, alarm pheromone producing organ of aphid is. Question number 8 is, what is poison baiting? That's all about today's class, guys. See you in the next class. Until then, bye.